Across warm tropics, papaya trees lift heavy green fruit toward rain and light. Beneath each unripe skin waits a living, milky white latex. To the tree, that latex is not waste but a protective flood. It beads when the surface is cut, sealing pale wounds in minutes. Inside the milky drops, papain rests among other quiet biochemical defenders. Its favorite material is protein, the long folded thread of living bodies. Protein gives shape to muscle, skin, seeds, milk, grain, and blood. But protein can also become a locked, stubborn, resistant material. A tough cut of meat resists the tooth, the knife, and time. A cloudy drink can hold tiny protein veils drifting in suspension. A hide can keep hair anchored by resilient protein roots. Nature faced this problem long before people gave protein any scientific name. Caterpillars bit leaves, fruit flies pierced skins, and microbes entered wounds. The papaya answered with chemistry rather than thorns, teeth, or claws. Papain helped loosen invading proteins, making each wound less welcoming to life. Long before laboratories, people noticed the tree's unusual softening power. In tropical kitchens, papaya leaves and green fruit softened meat for cooking. The discovery passed quietly through hands, markets, fires, and family meals. It was practical knowledge, learned by smell, touch, patience, and memory. Then the 19th century brought papaya sap onto disciplined European laboratory benches. European physicians and chemists were searching for new substances that aided digestion. They already knew pepsin from the stomach and diastase from malt. Papaya latex offered a stranger question from a distant tropical tree. In the 1870s, researchers studied its remarkable power to dissolve protein. Among them were Bouchut and Wurtz, working in Parisian medical science. They examined the milky sap of the Carica papaya fruit carefully. By 1879, they described a digestive ferment soon known as papain. The name carried the papaya tree into the language of enzymes. Papers and pharmacy notes spread the idea across Europe and the Atlantic. Here was digestion without a stomach, working inside a plant wound. That thought changed how scientists imagined the chemistry of living plants and bodies. Enzymes were becoming visible as active agents, not mysterious vital forces. Papain joined that new family with a distinctly earthy tropical origin. Its industrial story began with a simple harvest from unripe papaya fruit. Growers made shallow cuts and caught the white tears as they formed. When dried, the latex traveled as a concentrated botanical material for trade. Ports, pharmacies, and food trades carried papain into wider practical use. Early meat tenderizers drew on the same observation made in kitchens. Brewers and beverage makers valued its talent for clarifying protein haze. Leather workers explored it where hair and hide needed careful separation. Medicine also watched papain, attracted by its long and complicated digestive reputation. Some early preparations were crude, variable, and only partly understood by clinicians. Yet the central fact remained clear: papaya latex could cut protein. Later biochemistry revealed the small elegance hidden inside that old practical fact. Papain is folded like a tiny pocket made for molecular work. A protein chain drifts near, flexible yet strongly held together. The pocket grips a small section and bends it into position. At the center, reactive atoms pass a brief chemical charge. One protein bond loosens, then breaks into two gentler pieces. Papain releases them and waits patiently for another passing chain. It does not chew like an animal or burn like fire. It works by fit, nearness, and a single moment of exchange. Water finishes the cut, returning the enzyme to quiet readiness. This rhythm can repeat many times before papain finally becomes still. In food, the result can be a more tender bite. Muscle fibers and connective proteins relax where papain has gently touched them. In beverages, fewer protein hazes can mean a clearer, more stable liquid. In baking, controlled protein softening can change how dough stretches and handles. In plant-based foods, it can help create gentler, more pleasing textures. In leather preparation, enzymes can reduce dependence on harsher chemical treatment. In textile work, protein stains and fibers can be approached with unusual precision. In cleaning products, Papain can target traces from milk, egg, and blood. In research laboratories, it became a careful tool for studying the shapes of antibodies. Rodney Porter used Papain to reveal the architecture of protective antibodies. Through that work, an old fruit enzyme entered the story of modern immunology. Still, its deepest lesson remains visible on the papaya branch. A tree wounded by insects carries chemistry that can serve thoughtful human needs. When enzymes replace heat, harshness, or waste, industry becomes more attentive to nature. Papain reminds us that green technology may begin as a drop of sap, and the future may grow quietly, wherever life has already solved a problem. At Enzymes Bio, scientists and enzyme enthusiasts work to make this biology easy to reach. One platform, many enzymes, no tangled procurement, for research labs and growing businesses. A green technology with a long future. Learn more at enzymes.bio.